Assalamu alaikum. I am Dr. Sharoona and I am going to teach you the properties of the cardiac muscle today. Now, the objectives of this lecture are you have after the lecture you should be able to enumerate the properties of cardiac muscle. You should know what is the autorhythmicity of heart. You should also know the mechanism of excitation, contraction coupling in the cardiac muscle. You should know about the refractory period of the heart. You should know why it is important to have such refractory period and how it is produced. You should also be able to compare the action potentials of the SA node and the ventricular muscle.
और ये जब मैं क्लिक कर दूंगा तो उसके बाद ऊपर टाइम चलना शुरू हो जाएगा, रिकॉर्डिंग शुरू हो जाएगी। अगर पॉज करना हो तो सिर्फ इस पे जाएं और पॉज करें और
ये आएगा कैसे?
ये राइट क्लिक से नेक्स्ट क्लिक हो गया था, राइट क्लिक से। ठीक है, आपने सिर्फ राइट क्लिक दबाना और कुछ नहीं दबाना।
ठीक है,
राइट क्लिक से नेक्स्ट करनी है। राइट क्लिक जब आप कर देंगे ना तो ये पॉज हो जाएगा, पीछे रिकॉर्डिंग पॉज, सब कुछ पॉज। तो आपने सब कुछ राइट क्लिक से करना है। ठीक है, पॉइंटर आपका हर वक्त ऑन है। तब आपने जो भी कुछ करना है, अगली पे जाना होगा। पॉइंटर कहाँ चला गया? लेजर पॉइंटर आपका ऑन है हर वक्त। और अब आप एस्केप दबाएं एक दफा।
Now let's study the picture on the side of the slide. We see this is the cut section of the heart in longitudinal plane and this is the cross section of the heart. Here you see this is the left ventricle having red blood in it and this is the right ventricle having bluish blood in it. The wall of the left ventricle is thicker as compared to the wall of the right ventricle. Other important point in this diagram is that from the heart the oxygenated blood goes to different parts of the body and these blood vessels are in parallel to each other. The
हाँ, मतलब ये बहुत है, ये अगर सामने चीज लगी है ना तो कुछ नहीं, आप सिर्फ किससे कंट्रोल कर रहे हैं। अब आपने इसी स्लाइड पे ही रहना है क्योंकि मैंने पॉज कर दिया है, तो बस आप यहाँ एक दफा सिर्फ एस्केप दबाएं।
Okay, let's enumerate the properties of the cardiac muscle. These are the muscle is striated like the skeletal one. The muscle fibers are interdigitating with each other. It is arranged spirally around the circumference of the heart as you see it here. This is a spiral arrangement. The cardiac muscle fibers, if once dead, are not replaced. Now in the muscle fibers, mitochondria make 25 to 30% of the total cell mass, and this mitochondria is mainly involved in metabolism of the fatty acids for energy source. There is an extensive blood supply to the cardiac muscle, that is 1 capillary per fiber. The muscle cells have end-to-end anastomosis through desmosomes, and the gap junctions are present in the intercalated discs. You know of the gap junctions from your anatomy of the cell. I will be showing it to you in the next slide. One more thing, important thing to notice is that the cardiac muscle cell has got contractile elements, that is, the muscle fibers that contract to push blood into great vessels, and that is 99% of the muscle tissue. 1% tissue of the cardiac muscle is a specialized structure that is responsible for generating action potential in the heart muscle. It is the pacemaker tissue, and then this generated action potential is transported throughout the heart by a specialized fast conductor. As I said, this is a desmosome and this is a gap junction. Now, the single muscle fiber is 50 to 100 millimeter long and 10 to 20 millimeter in diameter. It is having end-to-end anastomosis, as you see here, and here. Now, this cell junction here is visible in this lower slide. The nucleus in the cell is central. Do you remember the nucleus in the skeletal muscle fiber? It was on the side of the cell along the cell membrane. Now, the contractile unit of the cardiac muscle cell is the same, the sarcomere, as in the skeletal muscle, having A band, I band, H zone and Z lines. Similarly, the sarcoplasmic reticulum is the same as in case of the skeletal muscle. The only difference is that the T tubules dip in at Z lines. The filaments present in the muscle are myosin, actin and tropomyosin, whereas troponin is the regulatory protein that attaches to the calcium ions in the muscle. Now, with this knowledge, you can understand that large number of mitochondria and fatty acid metabolism is essential for the cardiac muscle fibers, because every heart cell has to contract 24 hours per day, 7 days per week and lifelong similarly, whereas the skeletal muscle has a chance to take rest for prolonged periods. That is why the cardiac muscle gets so much blood supply and has so many mitochondria in it. Now, going for the study of the cardiac muscle, individually the properties are, it is striated, and this striation gives it a property of forceful contraction and enables it to pump out blood into the great vessels. Number 2, the cardiac muscle is not attached to the skeleton but makes a hollow viscus, and this hollow viscus accommodates blood and then it is pumped out by the muscle contraction. The contraction is again the same as the sliding filament theory of the skeletal muscle, but it is not under control of our will. The gap junctions in the muscle help the impulse action potential to travel from one cell to the other. And coming to the Frank-Starling's law, as you remember, Frank-Starling's law, in case of skeletal muscle, the force of contraction of the cardiac muscle depends on the initial length of the fiber. Here we see that the initial length of the fiber is actually the end diastolic volume that is stretching the fiber. Now do you understand? If the blood present in the heart just before contraction is more in amount, it will stretch chamber of the heart and the muscle will be lengthened. This will give a better contraction. If we have very little blood at the end of the diastole before the start of contraction of the heart, the muscle fibers will not be stretched and they will give a weaker contraction. And again we see that the skeletal muscle was having voluntary nerve supply and the cardiac muscle is having involuntary nerve supply. That is called autonomic innervation. The sixth property of the cardiac muscle is that it contracts as a syncytium. There are two syncytia in the cardiac muscle, one atrial and ventricular. Now can we go back to the physiology of smooth muscles? Do you remember that the visceral smooth muscle fibers acted like a syncytium? That is, a sheet of muscle fibers in the viscera will be contracting simultaneously to push the contents ahead, and they will be relaxing simultaneously afterwards. Similarly, the atrial muscle in the heart, the left and the right atrium, will be contracting at one time as a syncytium to push the blood into the ventricles. And after some delay, the ventricular muscle, the right and the left, will contract simultaneously as a syncytium to push the blood from the chambers of the heart, that is the right and the left ventricle, into great vessels. What are these great vessels? Anybody? Yes, they are the aorta and the pulmonary vessel. Coming to the next property, this is conductivity. The impulse is conducted through the gap junctions, as we have seen in the smooth muscle fibers. Now the eighth property is contractility of the cardiac muscle fiber, and like the skeletal muscle, it depends upon calcium. The source of calcium in case of cardiac muscle fiber is extracellular fluid as well as sarcoplasmic reticulum. The influx of calcium from the extracellular fluid depends upon nervous activity, certain hormones, and stretch of the muscle fiber. The ninth property is that the speed of contraction of the cardiac muscle is a little slow as compared to the skeletal muscle. Next property of the cardiac muscle is its long refractory period. The long refractory period in the action potential of the cardiac muscle helps the muscle to contract effectively and to pump the blood out of the chamber of the heart into the great vessels. This is because of the special action potential of the cardiac muscle, which is different from the skeletal muscle in the fact that it has got a plateau. After initial depolarization, there is a slight rapid repolarization, and then there is a plateau phase in which the cardiac muscle remains depolarized above the 0 level, 0 millivolt level, and then afterwards gets repolarized. This continuous positivity within the cell helps the muscle to keep on contracting because of presence of the calcium and avoids next impulse to come. Do you remember what was the reason for the refractory period? Do you remember the properties of the sodium channels that had activation and inactivation gates? Okay. If you remember, we know that the sodium inactivation gates can reopen at a voltage of minus 70 millivolts. So there is a long period of time in which the voltage does not reach minus 70 millivolts but is held above the 0 level, 0 millivolts, and that is the reason that the inactivation gates cannot be opened and second impulse is avoided. What is the advantage of this long refractory period? You know that the skeletal muscle has an action potential of 0.004 seconds and the contraction time is 0.1 second. And new action potentials can be received during the contraction that is occurred because of the first action potential. And this leads to tetanization of the skeletal muscle. The cardiac muscle has got an action potential of 0.3 seconds. There's a lot of difference between this 0.004 seconds and an action potential of 0.3 seconds. You see this difference? This was because of the plateau phase. Similarly, the contraction time is 0.3 seconds in the cardiac muscle, equal to the action potential time. So the contraction starts with action potential and no new impulse can be generated during the contraction time. This is the reason the cardiac muscle cannot be tetanized. And there is essentially a relaxation period in the ventricle after the contraction period. This allows filling of the chamber, and this is the objective of the heart. You have to pump the blood. If the ventricle is not filled, how will it pump the blood during contraction? So do you see the advantage of the long refractory period here? I hope you understand it well by now. Now we come to the detailed structure of the pacemaker tissue of the heart. Here you see in the picture there is an area of nodal area right under the receiving of the superior vena cava into the right atrium. There is another nodal area in the left wall of the right atrium here, and the two nodal areas are connected by these three pathways. This is brown colored is the contractile element of the atrium, here and here, all over, and red color is the conducting tissue in the heart. This blue color here, all these that we'll be discussing later on. So these are conducting pathways. Three conducting pathways are joining the SA node with the AV node. This is the SA node, sinus node, sinoatrial node, and this is the AV node, atrioventricular node, and the two pacemaker tissues are joined by these conducting fibers. You see, one fiber is going from the right atrium into the left atrium. This is called interatrial conducting tissue, and this is called intraatrial conducting tissue. From here, intranodal conducting tissue, it was intranodal conducting tissue between the two nodes, and this is interatrial conducting tissue joining the two atria and conducting the impulse from the SA node to the left atrium. Here, this straight blue structure is the bundle of His, and this is again a conducting tissue joining the AV node to the ventricle. This bundle of His is then branched on the endocardial surface. Are you seeing this? देख रहे हैं आप? ये जो है ये है एपिकार्डियल सरफेस, बाहर वाली ये इधर, और ये जो अंदर की सरफेस है इसको हम कहते हैं एंडोकार्डियल सरफेस. So the conducting tissue is first of all splitting into the right and left branches that are descending down the septum of the ventricles. This is the right ventricle, this is the left ventricle, this is the interventricular septum. After that, they are further branching and supplying the apex and the base of the heart, and these are the small branchings which are called Purkinje fibers. But filhal, at the moment, we are trying to see the SA node, and this is an ellipsoid cardiac tissue in the right atrium at entry of the superior vena cava. This node also is part of the cardiac muscle but has got very few myofibrils, the actin and the myosin filaments. Actually, it has got P cells in it. Along with that, there are transitional fibers at the periphery of this node. So the conducting tissue is going to join the contractile tissue by transitional fibers, and it has got nerve cells in it. These nerve cells are receiving nerve endings. Okay, here. Should we go ahead? Okay. Now, coming to the autorhythmicity, another property of the cardiac muscle, it is the inherent property of the cardiac muscle to generate its own rhythm. What do we mean by that? We mean that if we cut all the nerves coming to the heart while the experimental animal is still alive, the heart keeps on beating. It is not receiving any nerve fiber, still it is beating. It means that the nerve impulse is being generated within the heart, and this is the P cells in the SA node. They can generate this rhythm at a rate of 70 to 80 beats per minute, and in the AV node it is 50 to 60 beats per minute, and the AV node will generate the rhythm when this SA node is not working. And the ventricles, if they do not receive any impulse from the AV node, they are capable of generating its own rhythm at 30 to 40 beats per minute. Now we need to study this property of autorhythmicity of SA node or AV node or even the ventricular muscle in detail. At least this red line in the last of the slide, the property of autorhythmicity of SA node is due to its prepotential. This line you should remember because we make MCQs and even SCQs on the basis of this statement. So how is this prepotential generated? You need to know about certain ion channels, and first of all I will be showing you the ion channels responsible for the action potential of the SA node. What are they doing, what is happening, and what are the voltages at which these ion channels are opening or closing. Do you remember the ion channels of the skeletal muscle responsible for the action potential? Yes, you do. And now you see this black line at minus 60 millivolts, starting at minus 60 millivolts, depolarizing the membrane to a level of minus 40 millivolts. This black line is present in the SA node and in the AV node, and the action potential of the SA node and the AV node, I will be discussing the differences between the two, but at this moment this black line is produced due to number 1, opening of sodium channels that are different from the fast sodium channels in the skeletal muscle. These sodium channels open up when the membrane is hyperpolarizing. The membrane is coming down at the end of the action potential towards hyperpolarization, and it causes opening of the sodium channels. When was the fast voltage gated sodium channel opening? It was opening when the threshold potential was reached after depolarization. Do you remember that? If not, please, when you go home or when you stop the lecture, you can stop the lecture right here and go to the previous notes about the lecture on the action potential in nerves and skeletal muscle, and you will see that sodium channels, voltage gated fast sodium channels, open up when the membrane is depolarized to the threshold value. They open up here. But in case of SA node, the membrane is hyperpolarizing towards negativity, going towards negativity rather than the positivity, and here the sodium channels open up. Because this behavior is different from the routine fast voltage gated sodium channels, so they are called funny channels. So the permeability to the sodium increases through the sodium funning channels at minus 60 millivolts and allows sodium to go up. Here at this moment, when the voltage is minus 50 millivolts, the permeability to calcium through transiently opened calcium channels increases and the permeability through funny sodium channels decreases. P is for permeability. So now the rest of the black line is showing depolarization of the membrane slowly and gradually towards the threshold level, and this portion is from minus 50 to minus 40 millivolts is because of opening of the transient calcium channels, which will close at threshold potential. As the threshold potential is reached, that is minus 40 millivolts, long-lasting calcium channels open up to depolarize the SA node. Now, do you know what is long-lasting calcium channel? Have you heard of it before in the action potential of the skeletal muscle? No, they are not present in the skeletal muscle. They are present in the SA node. They are dependent on the voltage to open up, and this voltage is minus 40 millivolts. At this moment, long-lasting calcium channels open up. They are not opening as fast as the sodium, voltage-gated fast sodium channels. They are a little slow to open, so it is a rounded peak rather than the spike. They are long-lasting means they do not close as rapidly as the fast voltage-gated sodium channels, and because of that they are closing a little late, and this is producing this rounded peak of the action potential. And you see this rounded peak is a little above zero level. Remember, whenever you have to draw this action potential, and you will have to draw this action potential many times during your class, during your practicals, during your tutorials, during your vivas, and you have to make sure that you take the voltages in millivolts properly along the line, and you keep the peak of the action potential above 0 millivolt level. At this level, when the action potential is above 0 millivolts, what happens? The permeability of potassium increases through voltage-gated potassium channels. Do you remember how the voltage-gated potassium channels open up? Yes, they open up when a little positivity has been generated, that is, at the level of the membrane potential is above zero. It has to reach above zero, a little above zero. A little positivity is required to let these voltage-gated potassium channels open up, which will then produce repolarization. So this red line from here to here, up till here, is due to increased permeability of the membrane of the SA node to potassium. That was about the permeability, and here you see sodium is coming in slow depolarization. At this point, calcium is coming in, and this is slow depolarization. This black line is called the pre-potential. Bringing of the potential from minus 60 slowly to the threshold level produces self-induced action potential. It is not because of any nervous stimulus. Remember, potential is not due to any nervous stimulus. It is due to depolarizing pacemaker potential, inherent property of the SA node and the AV node. Now, as the action potential goes up, calcium goes in, and as the action potential goes down, potassium comes out, and this is self-induced action potential. And now we read it out on the slide. Slow influx of sodium and calcium ions drags the membrane potential to the threshold level, and an action potential is fired spontaneously. What is this? Remember, this is your exam question. Remember, this is your viva question, your tutorial question. Remember that this is the mechanism of generation of pre-potential in the pacemaker tissue of the heart, and the name of the pacemaker tissue is SA node and AV node. So mind you, we have taken a lot of time on discussion of the pre-potential, and now we go on to the ventricular action potential. That was the SA nodal action potential, and now we go on to the ventricular action potential, which has got 5 phases. There is a phase 0, and this is showing depolarization due to fast voltage gated sodium channels, giving it a spike. Next comes phase 1. There is a small spike due to transient rapid repolarization. Then the phase 2, voltage gated calcium channels L-type are responsible for plateau in the action potential. Phase 3, potassium channels and the leaky potassium channels are responsible for repolarization and also contribute in the plateau. Phase 4, ventricular muscle has no pre-potential, so phase 4 is absent. Action potential takes 0.25 seconds and starts after 0.16 seconds of the SA nodal action potential. The isoelectric resting membrane potential takes of the duration of 0.55 seconds is there, and this is present when the heart rate is 72 beats per minute. इसका क्या मतलब हुआ? अगर हार्ट रेट स्लो है तो रेस्टिंग मेम्ब्रेन पोटेंशियल लंबा होगा, अगर हार्ट रेट तेज है तो रेस्टिंग मेम्ब्रेन पोटेंशियल छोटा होगा. लेकिन एक्शन पोटेंशियल की ड्यूरेशन 0.25 सेकेंड्स की होगी नॉर्मली, और अगर हार्ट रेट तेज है तो ये भी थोड़ी सी छोटी हो सकती है. लेकिन रेस्टिंग मेम्ब्रेन पोटेंशियल हार्ट रेट के साथ ज्यादा वेरी करेगा, एक्शन पोटेंशियल हार्ट रेट के साथ कम वेरी करेगा. Now the sodium potassium ATPase revert back the ionic composition of the cell during resting membrane potential. ये भी बड़ा जरूरी है, इसके बगैर कॉन्ट्रैक्शन और रिलैक्सेशन ठीक नहीं होगी. Now we are comparing the action potential of the ventricle to the action potential of the SA node. Now it is the time for me to give you the phases of the action potentials. This is phase 0 in the ventricular action potential, that is the phase of depolarization, and this is phase 0 of the SA nodal action potential, that is the phase of depolarization. You have got a spike here, and after that there is phase 1. Phase 1, this is forming the spike in the ventricular action potential, and here there is no spike. You can say after the phase 0, phase 1 and 2 are not present in this action potential. This is the plateau phase in the ventricular action potential, and this is actually the phase 2 which is absent in this action potential. This is the phase 3 of the action potential, and that is the phase of repolarization, and here is the phase 3 of the action potential, and this is the phase of repolarization. This is phase 4 of the extranodal action potential, and it is usually absent in the ventricular action potential. This is the pre-potential giving pacemaker property to the SA node and the AV node, and it is absent usually in the ventricular action potential. Further comparison of the ventricular action potential to the SA nodal action potential, you see here fast voltage gated sodium channels open up to shift the membrane potential from minus 90 to minus 70 and then to plus 30. Here the sodium current suddenly decreases and potassium comes out fast through transient channels. Now the calcium channels open up, calcium is going into the cell and potassium is coming out of the cell, so the plateau is formed here. Positive charge is coming in, calcium, potassium going out, positive charges go out, and this produces an plateau in the action potential of the ventricular muscle. Now potassium starts coming out through fast voltage gated potassium channels and the action potential goes down to baseline.